In the previous two videos of the cell cycle, we studied about the interphase and we studied the M phase or the mitosis in detail. Mitosis was a division where the number of chromosomes in the parent and the progeny were same. Before you understand meiosis, you should know all the stages of mitosis, and therefore I am going to link that video in the description, and you can also find it in the corner tab of this video. Watch that video first and then you can start with this one. The two concepts that we are pretty familiar with are addition and the level of population we have in our world. So, according to the fertilization and fusion of the gametes, if one of the gametes has, say, 2 chromosomes, or the parent or the father has the 2 chromosomes, and the mother also has 2 chromosomes, and these two combine, the child will end up having 4. The number of chromosomes in a human cell are 46. If these combine, the child will end up having 92 chromosomes. And now imagine that if there are so many people in the world and all of these divided this way, our cells won't be able to sustain themselves. There will be an extremely large number of chromosomes and none of them will eventually express themselves. This is the reason why we have one very important type of division, that is the meiotic division, which is also known as the reductional division. If you remember, mitosis was known as the equational division because the parent and progeny has the same number of chromosomes. In the reductional division, the parent will give half of its chromosomes to the gametes. So if the parent in a human being has 46 chromosomes, only 23 of them will be transferred to the gamete. And when both the gametes combine during fertilization, the progeny will again have 46 chromosomes and therefore it will be able to sustain itself. This cell with 46 chromosomes will divide again and again and finally give rise to a life-size human being. This division is mitotic. So, meiotic division is very, very important when we have to reduce the number of chromosomes in an individual to half. Meiosis is also divided into 2 different phases. The first one is meiosis 1 and second is the meiosis 2. They are pretty easy names for a change. Meiosis 1 is again divided into the PMAT that we studied about, the prophase, metaphase, anaphase and telophase. The prophase 1 is a very detailed one. Metaphase, anaphase and telophase are pretty much the same as they were in the mitotic division. Meiosis 2 is entirely same as mitosis. One characteristic of meiosis is the presence of homologous chromosomes and the recombination between them. Homologous chromosomes are present in the body in pairs and the recombination is the exchange of the genetic material. You will understand both these terms as we study about the prophase 1 in detail. The prophase 1 of the meiosis 1 is divided into 5 different phases: the leptotene, zygotene, pachytene, diplotene and dikinases. At the end of the prophase, as it happened in the mitotic division, the cell will not have a nuclear envelope and all the organelles that will be present in the cell will disappear. Along with this, at the end, the spindle will form. Now in these 5 phases, what happens is that the homologous chromosomes come together, exchange their genetic material in the process of crossing over, and at the end we have a variation of the chromosomes that were initially present. So this was a brief introduction to meiosis, what it is and why is it needed in our body. In the next part, we are going to start with meiosis 1 and the prophase 1 of meiosis. Prophase 1 is pretty detailed and we are going to understand that meiosis 2 and the significance of meiosis in the next part. Hope you understood whatever we did today. And if you like this video, don't forget to press the like button and subscribe to my channel for getting future updates of the next parts. Thanks for watching.